Hi, good evening. So we will start today's session. Just a minute. Just give me a minute. So we will start today. We'll start sharing the screen now. Is it visible? You guys can unmute yourselves and let me know. Just yes. So today we will summarize week 7 lecture content and then we'll look at some sample questions. Okay. So, this week's video mostly consisted of all how would you really break open the cells? What are the different cell disrupting techniques that one can uh, perform so as to break open the cells and take the contents out, okay, for different kinds of downstream processes. So, disruption is a process where we can obtain the intracellular material or fluid via methods that actually break open the cell wall if the cell has one, without disrupting any of its components. Okay, and cell disruption techniques can vary depending on the type of cell and its cell wall composition, because plant cells, bacterial cells have different kinds of, as well as fungal cells have different kinds of cell wall composition. And definitely, we should be careful that the cell disruption technique that we are undertaking shouldn't be too harsh, so that the product that we want to really take out, whether it be it nucleic acid or proteins or any other content or lipid component, that should remain intact. The intracellular material that we want to take out of the cell should be intact. And there can be different mechanical methods that can be divided very broadly into solid shearing methods and liquid shearing methods. And there can be non-mechanical methods as well, which can again be divided into physical methods, or cells can be treated chemically, or chemically using enzymes or some other solvents, etc. Okay, so we will look at each of these methods in detail. So this is what is the flow chart of the complete classification of different categories. So one is using mortar and pestle, where one grinds the cells so as to disrupt the complete cell and break it open. And for using mortar and pestle and grinding the cells needn't be in a suspension, right? In a liquid culture. It can also be usually yeast cells or plant cells. What people do is they have a cell pellet, the cells are pelleted, and or even the suspended cells are actually frozen in liquid nitrogen. So you have balls of frozen pellet cells. Okay. And that is actually crushed or grinded using mortar and pestle. And then you actually treat that grinded cells using different kinds of buffer or liquids, etc. Okay. And so that is what is written here, that for mortar and pestle disruption, the cells needn't be in suspension. It is often done with samples. It can be fungal samples as well. It can be, mostly it is done with plant samples. It is frozen in liquid nitrogen. Otherwise, it's very difficult for the tough and robust cell wall of the plant cells to break open. Okay? And metabolites can be extracted from the disrupted material by adding different kinds of solvents. Whatever is the appropriate solvent or buffer of your interest, you can add that and then extract your product of interest. Then there can be blenders or the mixer grinders. Simply what we use in day-to-day kitchen activities, what we use it in day-to-day kitchen activities, those blenders can also be used at high speeds at a for a certain period of time to disrupt the cell walls. This is also done with plant samples frozen in liquid nitrogen. It is the same process when once after centrifugation, it separates or concentrates the materials which are suspended in a liquid medium. Then there's bead beating. All of this we looked at very briefly in previous tutorials as well, where there are glass or ceramic beads which are used to it's vortexed or centrifuged so that the glass beads or ceramic beads uh, interact with the cells in suspension and break it open. Okay, it's gentle enough to keep the organelles intact. So we have to be mindful of the centrifugation speed, the speed of vortexing, the time till up to which we are vortexing the complete sample. And bead beating method can be used with all kinds of cells. What basically we have to do is the cell suspension is there. Just add the required amount of bead to that particular cell suspension and vortex or centrifuge. Okay. Or even with samples which are like really we have to be really gentle and very careful while handling those samples. In those cases, what one can also do is vortex rather than vortexing. One can also do is simply rotate it at a in a rotator. Like it's a rotator where you can attach your tube with the cell suspension and the glass beads and let it rotate. Let the sample rotate. So what will happen is due to the rotation, 360-degree rotation, complete rotation, the glass beads that are there in the sample along with the cell suspension, those will actually, uh, constantly collide with the cells and disrupt the cells, break open the cells and let the contents out or intracellular contents out of the cells. So this is a 2023 study. You can always go back and read this. So this is taken from there. So suppose there's this particular cell that needs to be disrupted. So what happens here is these black ones are the beads and the red ones are the cells of our interest that needs to be disrupted. So what happens is um, it is getting centrifuged or rotated in a 360-degree rotation. So what is happening here is the beads are colliding between with the cells, and then there can be three different kinds, like mainly broadly three different kinds of force. One is the shearing force, like between two beads there is enough gap so that the cell wall gets out, but the intracellular contents, intracellular molecules can come out of the cells. Okay. Or there can be impact force. Definitely the beads are actually directly hitting the uh, directly actually colliding with the cells, individual cell, and then basically the impact is actually uh, the force, the impact force is actually breaking the cell open, or there can be direct collision which can also break the cells open. Okay, so this is the study where they actually summarized this bead peeling method. You can go through it in detail if you want to, like if you're interested. And then there's sonication. Now, sonication is again a mechanical method to break open the cells, where the homogenizers, ultrasonic homogenizers, actually work where vibrations are induced in a titanium probe that is immersed in the cell suspension or cell solution. And then there is this thing called cavitation. This is a phenomenon where tiny bubbles are formed and unequal sized bubbles are formed, which explode, and local shock waves are sent across the cell suspension. And due to these shock waves, pressure changes occur and it disrupts the cell wall and let the intracellular, like lets this sonication process lets the intracellular content out of the cells. Okay. And this is sonication is used for bacterial cells, plant cells, fungal cells. Okay. So this is the titanium probe we were talking about earlier. So when you have to provide a certain uh, frequency where this electric probe supplies the sound energy, which gives rise to an unstable growth of cavitation bubbles, which leads to unequal sized droplets. And these droplets eventually burst open and local shock wave is produced, and which further disrupts the cell walls. So this is what is happening here. Bubble formation takes place. The sonic waves are going having at a particular amplitude. This amplitude is variable. The user can set the particular amplitude, and due to expansion compression as the wave travels, these bubbles are formed. Unequal like unequal sized growth bubbles are formed. Cavitation bubbles are formed, and then suddenly the bubble explodes, thereby disrupting the cell walls nearby. Okay. Then there's another method called homogenization. It is a liquid waste homogenization. Again, this is another technique to disrupt cells for small volume cultures, where the cells are actually lysed by forcing that particular cell or tissue suspension by passing those cells or suspension through a narrow space. And then the shearing forces on the cell is applied. It is similar to the bead beating method or bead milling method, where we saw that the complete complete cell cannot pass through the small space and the cell wall is retained back and the intracellular content comes out. Okay? Basically, we are squeezing the cells through a tube that is actually smaller than the dimension of a cell. Okay? Then there's freeze thaw method, where uh, basically usually it's for soft tissues, soft plant material, algal cells. Okay, where the cells are disrupted by a series of freezing and thawing cycles. So what happens is when you are freezing forms ice crystals, which upon when the sample is thawed or it melts, it expands eventually, and the cells, especially the cell walls, outer layer of the cell walls, rupture. Okay, due to multiple freezing and thawing cycles. Then one can also do thermolysis, basically exposing the cells or cell suspension or tissue suspension to a high temperature. Okay, or one can even microwave. So microwaving along with autoclave and other high temperature methods can actually disrupt the bonds within the cell walls that are actually holding the cell walls, like maintaining the integrity of the cell walls. Basically, high temperature can also denature proteins that are like constituting the cell walls or the cell membrane. Okay. So, but the exposure to high temperature methods have to be really controlled, because uncontrolled amount of heat can actually easily denature or damage the target proteins and substances. Okay. Then there's osmotic shock as well. Basically, the phenomenon of osmosis is taken care of. Like we place the cell in a suspension with hypotonic solution where the concentration of water is really high as compared to the intracellular water concentration. So the water actually moves in, thereby the cell swells up and bursts open. Okay. So water moves into the cell, causing its volume to increase till a point such that it can burst open. And this osmotic shock can actually work with animal cells and protozoans, since they do not have cell walls. Otherwise, only using water to get like only using osmotic shock to break the cells open for the cells having cell walls, it's a bit difficult. So usually osmotic shock actually works for cells that do not have a cell wall. Okay. Now most of the mammalian cells have a plasma membrane with an active transporter protein that maintains the osmotic balance. And when an osmotic balance is maintained, it is actually an active process which requires energy. Okay. So when these mammalian cells are exposed to hypotonic solution that I was talking about, having very less concentration of solute, like more water concentration. So there is an osmotic imbalance which ultimately lyses the cells, because water moves in, the cell swells up and disrupts. So there's this Hoff's equation where the osmotic pressure can be calculated, and which has been found to be directly proportional to the concentration of the solute as well as the temperature. okay, which is pi is equal to RT and the concentration gradient, the initial and final concentrations of the total solute inside and outside the cell. Okay. And also each type of mammalian cell has actually a differential susceptibility towards osmotic shock method. Now, for example, red blood cells. These can be lysed by adding a drop of water. But plant or bacterial cells, which actually have a robust cell wall, these are more resistant towards osmotic lysis method, because they have a thick cell wall. Okay, but red blood cells, which only have a cell membrane, these can be lysed easily using osmotic shock method. Now, there can be electric discharge method as well, where the cells can be disrupted by electric discharges, especially in mammalian cells that are bound by plasma membrane. Only plasma membrane that do not have a cell wall. Okay. Then there's chemical lysis methods, where you are basically disrupting the cell by treating the cells with different kinds of solvents or chemicals. There can be organic solvents like alcohol, ether, or chloroform, which actually disrupt the cell wall by permeabilizing the cell walls and membranes. Okay, so even for nucleic acid extraction or RNA extraction, what one does is usually one treats the cells with chloroform, which creates pores inside, and it takes out the contents of the intracellular content of the cell. So chemical lighting method is actually very handy and very useful in extracting hydrophobic molecules like plant pigments. Also, one can use a chelating agent like EDTA. So EDTA chelates divalent ions, right? So many divalent ions are actually important to maintain the integrity of the cell wall, like magnesium and calcium. So if a chelating agent like EDTA is used, it can specifically disrupt the cell walls of gram-negative bacteria, because the cell walls of gram-negative bacteria actually contain lipopolysaccharides that are actually stabilized by these divalent cations, magnesium and calcium. And then once the cations are chelated out, these ions are chelated out, it destabilizes the cell wall integrity, and the cell wall can be ruptured easily. And later it has to be dialyzed away. Mainly the EDT has to be removed, because suppose if you wish to isolate protein, purify protein, or if you wish to purify nucleic acids, or if you want to purify ribosomes, etc., what actually happens is these cations are actually very important as well in maintaining the complete functional structure of a macromolecule, right? Even for proteins or nucleic acids or a ribosome. Okay, the interactions with these divalent ions are actually necessary to maintain these macromolecules or macromolecular machineries in a functional state, right? So what happens here is, once you have used EDTA to disrupt the cell wall of bacterial cells, and for downstream processes, it is a good idea to always remove the EDTA completely, so that the EDTA does not later affect or interfere with the downstream purification processes. Whichever product you want to purify, this should not actually interfere with the downstream processes. Then there's enzymatic lighting methods, which actually breaks open the cells using enzymes that degrade cell walls and membranes. So depending on the type of organism, one can use cellulases, chitinases, or lysozyme enzymes, bacteriolytic enzymes, okay, which lysozyme actually destroys the peptidoglycan. And also enzymatic lysis can be really useful if you want to isolate the protoplast, right? And what is a protoplast? This is basically a cell without the cell. Okay. Another thing that can actually disrupt the cells is alkali treatment. This is actually a harsh cell disruption technique, but it is an effective chemical treatment to break the cells open. And what actually happens is, when you treat the cells with alkali, it actually leads to lipid saponification, which mainly disturbs or disrupts the lipid packing and affects the cell wall integrity. Also, one can add detergent when you treat the cells with a detergent solution, that can also solubilize the lipid that mainly constitutes the cell membrane, the plasma membrane. And also the effect of detergent on cell wall has been found to have a linear correlation with its concentration. Okay, why? Because the detergent concentration can cause a sudden change in the solubility of lipid and also leads to the formation of micelle, right? And there can be different kinds of detergent that can be used like CTAP, Tritonex, Saponin, SDS. SDS, we looked at SDS in SDS page in the previous tutorial as well. And then there's permeabilization. What happens here is a partial cell wall disruption or permeabilization can be uh, done or achieved by using an organic solvent like toluene. Okay? And what happens here is when you are treating the cells with an organic solvent like toluene, so this organic solvent is getting absorbed by the cell wall, resulting in the swelling of the cell and ultimately rupturing the cell. But when you're actually using the solvent at low concentration, the solvent can actually permeabilize the cell wall without disturbing the cellular integrity. So this concentration actually has to be optimized. And then this method actually allows to use the cell as a reaction vessel to catalyze any reaction and get the desired product or purify the desired product. So basically you have ultrasonic extraction where ultrasound is used to create vigorous agitation resulting in the extraction of the analyte from the matrix. Analyte is basically the product of your interest. And there can be mechanical shaker extraction where this it depends on the selection of the solvent in which the solubility of the analyte is higher. Basically, the product that you are wishing to purify, you wish to purify. If you pass that or you expose mixture of the suppose your product is X and X reacts the most or is most soluble in a solvent Y. But along with X, you have many other different kinds of molecules like A, B, C, Z, uh, like that, all different kinds of mixture is there. But if you expose this particular mixture or pass this particular mixture through the solvent Y, to which X has the like in which the component X has the highest solubility, then what will happen is X will dissolve first and it can be extracted out of the complete mixture. Okay. Similarly, there can be liquid extraction as well, where basically you are partitioning the sample between two immiscible, the liquids which do not mix between each other, which doesn't dissolve. So between two immiscible liquids, you actually are partitioning your sample or your analyte in the extraction of interference, free analyte into the liquid in which the solubility is higher. Two immiscible liquids are there and your product of interest, which can also be called as analyte, that will either go into one of the liquids and you can eventually take that liquid out and further extract your analyte from that particular liquid. Then there can be salting out, solid phase micro extraction, etc. as well. Each of these we will look in the next slides. Okay. Now, how two molecules can be separated from each other? Suppose there are three molecules, benzene, phenol, aniline, and these are actually very similar to each other, but they have different physical and chemical properties. And these differences can actually be exploited to separate these molecules from each other. Okay, the physical properties can be the molecular weight, the boiling point. Okay, when we are talking about when the analyte in question is a liquid and the complete mixture is a liquid as well. So we suppose the liquid that has a lower boiling point or a higher boiling point, the one with the lower boiling point will evaporate like based on the time points when one liquid is evaporating faster than the other, we can actually separate out the two liquids. Okay. Freezing point or crystallization, solubility, as well as the density. And then chemical properties that can be exploited is the functional group. For example, phenol has an OH group, whereas aniline has NH2. And another thing that can be exploited or used to separate these separate your analyte from a mixture is the reactivity towards other agent to form a complex. So this is an example where you have a mixture of these two compounds, benzene and aniline, suppose. Okay. And you would like to separate benzene from aniline and you are mainly interested in taking out the benzene from there. So what you can do is One way to do is benzene. Suppose you are boiling the liquid, benzene is evaporating faster, then you have a condenser attached here. So eventually the benzene will actually condense here and the benzene can be collected. So aniline is there in your flask already and benzene can be collected in a separate container altogether. Eventually you have two separate liquids separated from each other, nicely separated from each other. Okay. So when we talk about the distribution coefficient, is basically the distribution of this compound 1 or molecule 1 between two phases A and B, and Kd can be concentration in phase A divided by the concentration in phase B. Okay. Now we come to chromatography. So what exactly is chromatography? Chromatography is used to separate a complex mixture into individual components. How? Based on the partitioning effect, which distribute the mixture of molecules into different phases. Okay. The distribution of a molecule between two phases, suppose a stationary phase, a mobile phase, a liquid phase, is given by the distribution coefficient that we looked before. Okay. Usually the two phases, out of the two phases, phase A is a stationary phase or the matrix and phase B is a mobile phase or the buffer. Okay. So actually chromatography was invented by M. Sweat. Basically what the person did was on a matrix of calcium carbonate, uh, chlorophyll was loaded and the solvent ether was passed. Okay. And when chlorophyll pigment was loaded and ether solvent was passed, so on the matrix of calcium carbonate, the chlorophyll pigment actually separated into different colors. So that's where we get the name chromatography. Chroma refers to colors. Okay. And Sweat actually invented the method of chromatography. He actually performed chromatography for the very first time. Okay. Now, when we talk about column chromatography, what happens in column chromatography? There's a stationary phase or a matrix. Okay. And a mixture of analyte is loaded here. This particular sample mixture is having is consisting of, for the sake of simplicity, only two kinds of molecules are there in the sample mixture, the red ones, red squares, and these starry yellow stars. Okay. Now this mixture or the sample mixture is actually loaded on the top, and then based on the porosity of the matrix as well as the size of the molecules in the sample mixture, it will eventually get separated, and the separated molecules can eventually be collected separately so as to get your product of interest. Now, suppose these red squares are there and yellow stars are there, right? So these are two molecules with a Kd value, distribution coefficient value of 1 and 9, and they are actually traveling through a column with water as a mobile phase. Now, if the red cubes, the red squares are more soluble in water, this is a stationary matrix and water is passing through it. Okay. Now, if the red squares are more soluble in the water, then what will happen is it will eventually separate out and mix readily with the water phase. Okay. Water that is being passed through the matrix. And the yellow stars actually have more affinity or rather less affinity to water or more affinity with the matrix. So it will eventually move very slowly, but the mobile phase water is moving rapidly. So the one which is more soluble in the water will move much rapidly. In this case, the red squares. Okay. So as the mixture will travel, it will partition between the stationary phase and the mobile phase. But because there's a huge difference between the Kd values, so if the value is lower, Kd value is lower, it will have very less affinity to the matrix or stationary phase. Okay. It will have a higher affinity with the mobile phase, and it will eventually be eluted out first. Okay. So, or rather, it will come out first, followed by the one that has a higher Kd value. Okay. Now, what are the major parameters that one should consider while preparing a sample for chromatographic purification? One is clarification. You have to really clear your lysate or sample from any kind of cell debris. Like you have basically disrupted the cells and taken out the intracellular content, right? So you have to clear the sample from any kind of cellular debris, etc. Then you have to stabilize the target protein, like target product rather. So if it is a protein, then you have to stabilize it in a functional form, in a properly folded form. There should be, suppose if you are talking about protein, then we should be having protease inhibition. Any kind of like action of proteases should be avoided. We have to maintain a proper pH, definitely, because we were looking the previous tutorials how the surrounding pH of the medium affects the overall charge on an amino acid and hence the complete sequence of amino acid. So the pH of the solution or the buffer has to be taken into account. What are the different reducing agents in the solution? Okay. What are the different stabilizing additives that have been added? And then what are the conditions for purification to work? Mainly adsorption, how much amount or how much amount of buffer, how much amount of sample has to be added for it to properly bind to the matrix. But if we are talking about that molecule of interest is a protein, then we have to be really careful and optimize it properly so that only our target protein is binding to the matrix. And the contaminants. There can be, like when you are lysing the cells, complete protein content of the cell is huge. There can be n number of proteins. But out of those crowded milieu, you have to take out your protein of interest. So basically, you have to minimize, you have to optimize such that only your target protein is binding and contaminants aren't binding there. Okay. And definitely, what are the available resources in your laboratory, in your experimental area, or your institute? That actually matters a lot in terms of what purification technique you will be applying. And definitely, what are the practicalities to be considered, the sample size, like how much, whether you need 1 ml of protein, whether you need 1 microliter of protein, that also makes a huge difference of how much protein you need to purify. Like I'm taking the example of proteins, but it can be any other thing, lipids, mitochondria, ribosomes, etc. Now, centrifugation and filtration are actually standard laboratory techniques for sample purification. I was talking about removal of cell debris, right? Uh, these are actually routinely used when handling small samples. Centrifugation will basically separate out the cell debris, will pellet down the cell debris, and the supernatant, you basically have to process the supernatant later. And filtration, you basically, how you do is you pass your cell suspension through a filter size of having different kinds of pore sizes. So the smaller the pore size, usually zero point 0.2 micrometers, zero point 0.4 micrometers, zero point 0.5 micrometers. There can be different pore sizes, filter membranes having different kinds of pore sizes. So uh, suppose if 0.22 micrometers pore sized filter is being used, so the any kind of debris or any particle above having a dimension more than having a diameter more than zero point 0.2 micrometers will be uh, like filtered out of the complete cell suspension. Okay. Also, one needs to be very careful of keeping the samples on ice until use, which is often recommended even when purification is performed at room temperature. So what centrifugation is doing is it is removing most of the particulate matter like cell debris, as I was talking about earlier. Filtration also helps removing particulate matter like membrane filters that actually give the least amount of non-specific binding of proteins. Also, when you are actually filtering, one should be very careful of the type of filter membrane that is being used. Okay, because it has to be a type of membrane that the your molecule of interest that you want to purify or isolate shouldn't be interacting with that particular filter membrane. Okay, then only you can actually eventually take it out. So membrane filters that give the least amount of non-specific binding of proteins are composed of, usually the type of filter membranes that are used is PVDF, polyvinylidene fluoride, or cellulose acetate. There can be nitrocellulose membranes as well. Okay. And also before chromatography for sample preparation, the filter pore size in relation to the bead size that are actually used to pack the chromatographic column, to pack the matrix, should be selected. We have to be careful about what type of molecule we want to isolate and purify. Okay. One needs to be careful about the protein stability as well. So in majority of the cases, when we are purifying the molecule of interest, macromolecule, with be it ribosomes, be it nucleic acids, be it proteins, or uh, so these macromolecules have to be have to be isolated in a way such that the biological activity is retained, because ultimately what we are interested in is looking at its biological function. So ultimately, suppose if we are purifying a protein, the protein should be functional after the complete step of purification. So retaining the activity of the target molecule is actually important while following the progress of the purification, and detection of the target molecule also often relies on its biological activity. So many times the proteins can are easily prone to degradation or denaturation, and it's not really stable uh, at room temperature. It has to be always maintained in a on ice or at 4 degrees Celsius. So all these things have to be taken care of. Okay, and it can some samples can easily denature like RNA, right? RNA or ribosomes. Denaturation of sample components have to be taken care of. Sometimes samples can be are prone to aggregation as well, like many of the proteins having disordered regions in their uh, sequence, disordered regions. So what happens is it leads to precipitation or aggregation. So this has to be taken care of as well. So many times while purifying proteins which are prone to aggregate formation, it actually precipitates or can lead to non-specific adsorption on the matrix, which eventually impairs the function of the column or the separating or resolution of the column, separation ability of the matrix in question. So there are many advantages to checking the stability limits of the sample you are working with and working within these limits during purification. Okay. Now, proteins generally contain a high degree of tertiary structure, mostly due to the van der Waals forces, hydrogen bonding, ionic and hydrophobic interactions, right? So any condition that is capable of destabilizing these forces, or any condition that will affect or destabilize these forces, can lead to denaturation of the protein, degradation of the protein, and the protein can eventually precipitate out. Now, peptides have low degree of tertiary structure, and their native state is usually dominated by secondary structures, and secondary structures are mostly consisted like secondary structures mostly consist of hydrogen bonding. Now, peptides can tolerate actually a much wider range of conditions than properly folded proteins. And proteins are actually once denatured, it is not easily renatured, while peptides can often renature spontaneously. Now, when we talk about the quaternary structure of proteins and the protein complexes, this becomes much more challenging as compared to only proteins or peptides. Now, when we are talking about complexes, protein complexes, which are often held together by really weak interactions. So in that case, we have to be careful about the purification conditions. It shouldn't be too harsh so that we are actually disrupting the interactions that are holding the complete complex. So that has to be taken care of when we are purifying the complete complex. Also, some many times we have to be careful about because we eventually want suppose we want to purify a protein, and the protein should eventually retain its biological activity, and for the protein to have a uh, like functional reaction, to be functional, for the protein to be functional, basically many times cofactors or coenzymes are required, right? So that has to be also taken care of in the buffer condition, or while performing any kind of assay for that with that particular protein of interest. Then there are membrane proteins that are altogether a different challenge in itself. Purification of membrane proteins and studying membrane proteins is itself a whole different challenge. So membrane proteins may also need lipids from their natural environment to maintain their native structures, right? So it's usually actually advisable to perform stability tests, what we were talking about, before beginning to develop a purification protocol. Now, whether the protein or your product of interest that you wish to isolate or purify is prone to aggregate formation or prone to aggregate formation, that can be checked using different kinds of chromatography techniques, mainly size exclusion chromatography, to assess the uh, aggregate formation tendency of the protein or the molecule in question. We will look at SSC in great detail in the later tutorial. Also, one needs to check if which range of pH is actually more optimum for the molecule to stay to be stable and to stay functional. Then also how much salt concentration, we have to be really careful about the salt concentration as well as the temperature stability. I was talking about some proteins actually denature very easily at room temperature. So it has to be always maintained uh, in cold conditions. Okay. Then one has to test for the protein stability and proteolytic activity. Okay. And to assay for the proteolytic activity as well, we looked at SDS pH. So one can always check the integrity of the protein, purity of the protein on an SDS pH as well. Now there are few terms to be um, there are few terms which you might come across eventually while reading about chromatography. One is the chromatogram or the chromatograph. Then there's eluent, the fluid or the mobile phase that is, or the solvent that is entering the column. Eventually the analyte is there. Then eluate is the mobile phase that leaves the column or the matrix in question. Stationary phase is basically the immobilized phase. Uh, these are actually immobilized on the support particles or on the inner wall of the column tubing. For example, there's silica layer. There can be different kinds of stationary phase or matrix. Okay. Then mobile phase, we are talking about the mobile phase basically moves through the column or the stationary matrix where the sample can interact with the stationary phase and is separated. Then there's another term called retention time, which is actually the time required for a particular analyte to pass through the system under the given buffer conditions or pH conditions, etc. And sample or analyte is the substance that you wish to purify, and solvent is any kind of substance that can solubilize another molecule or another substance. Now then there's chromatogram. This is actually the visual output of of chromatography, and usually you see the separation as different peaks or patterns on the chromatogram. This is basically a chromatogram, uh, referring to the different components in the complete mixture. Okay. So usually it is represented in a graphical format where the x-axis, the independent variable, is the retention time. Either it can be retention time or the retention volume. Okay, volume is basically the mobile phase constantly keeps on moving, right? So at what volume of the buffer or the elution buffer, your sample of interest is coming out or getting separated. Y-axis is basically the signal. So suppose when I'm talking about proteins, now there are proteins are composed of amino acids, and there are certain amino acids like the aromatic amino acids, tryptophan, uh, phenylalanine, and there are these aromatic amino acids which can absorb UV, which get the due to the electron density, diffused electron density, it can actually absorb UV and get excited and give out signals. So based on the absorption of these amino acid, how the signal is given out, the signal is proportional to the concentration of the specific analyte that is being separated out of the complete mixture. Okay. Now, so what can be the different factors that affect the separation of a complete macromolecular mixture? What happens is the intermolecular interaction between the two phases, mainly the stationary phase of the matrix in question and the mobile phase that is passing through it, and the extent of dispersion of solute molecules over the stationary phase. Okay. So this is the mobile phase. The solute is transferring from the stationary phase to the mobile phase at the peak of at the back of the peak profile. So you will get peaks eventually. and one peak will eventually refer to a particular set or a single uh, analyte in question. Okay, and two processes actually occur on the column, like on the matrix. Number one, what happens is the peaks are moved apart as a result of their relative affinities for the stationary phase. The peaks get separated based on how much is the interaction between. So if uh, the this distribution is well resolved, then that means suppose there are three components in the sample mixture. So these three components have like distinct affinities with the matrix. Or if there are overlapping peaks here, that means it is not getting properly separated out or properly resolved on that particular matrix in question. And the spread or dispersion of the peaks is constrained so that the solutes can be eluted discretely. Okay. So there is this term called resolution. In chromatography, we actually often use the term resolution, which refers to the selectivity of the resin or the matrix in question, and how efficient is that matrix or resin in producing a sharp, narrow peaks with minimal peak broadening. Okay. So actually, there's a formula as well to express to quantify the resolution of a particular matrix, which is if there are two peaks, well separated peaks, okay, two adjacent peaks, the elution volumes are actually the difference in the elution volumes serves as the numerator upon average of the width of the peaks. Okay, uh, width of the peaks is used so as to quantify the resolution of a particular matrix. Now, in general, when I'm talking constantly about a chromatographic matrix, so what exactly happens there is a particular matrix or a chromatographic column is made up of multiple plates. Okay, so in chromatography, a theoretical plate is a hypothetical zone or a stage in the column, basically small regions or zones. Okay, where the solute components achieve or the analyte mixture or sample mixture achieve equilibrium between the stationary phase and the mobile phase. Okay, so basically this concept has been taken from distillation theory and adapted to chromatography so as to measure the efficiency of separation. So this theoretical plate, the idea of theoretical plate that consists of a like constitutes a chromatographic column efficiency can be uh, talked about. So what is column efficiency? The number of theoretical plates that a chromatographic column contains. So if there are a higher number of plates, the higher the number of plates, the better separation efficiency that particular matrix can have, the column can have, chromatographic column can have, because the solute is undergoing more equilibration steps at each of these zones, right? So hence the separation will be much sharper and much more distinct. Okay, the separation of the each of the solute in the sample mixture will be more sharp and more distinct from each other. Okay, so this is what is here. If there are more plates, a sharper peak will be there. If there are less plates, a broader peak will be there and less resolution will be there. Okay. Now there are multiple, you can, like I will be sharing this PPT as well, where there are multiple terms which you can come across. So we looked at most of this. Basically, we have been talking about adsorption of the solute particles or sample on the matrix. Basically, adsorption is the binding of the solute particles where it interacts with the stationary phase. Okay. Affinity chromatography is the different kinds of group of methods which actually exploits the types of specific affinities between the target molecule, like a protein, and a specific ligand which is associated or attached to the chromatographic matrix. Then there's band broadening, the widening of the zone of solute. We looked at sharper peaks or broader peaks. That exactly is band broadening. Okay. So if when passing through a column or a chromatographic matrix, if the peak actually is broadening, if the peak is widening, band is broad, which means solute has been diluted and resolution of the matrix, resolution of the column is really low. Okay, this band broadening is also known as peak broadening or zone broadening. Binding and adsorption are can be used interchangeably. There's binding buffer, which is actually used for the equilibration of the column before loading your actual sample. Because your sample, suppose that you want to purify a protein of interest, and that protein should have a proper pH so that it can have the appropriate charge, overall net charge on it. So, for it to have the appropriate conditions, for it to remain stable in the solution or on the matrix as well, we have to provide uh, same or similar conditions on the matrix as well, so that the protein can eventually bind or elute out of the matrix but still remain stable and correctly folded. Okay, if the molecule of interest in question is a protein. Then there's chromatogram. We looked at chromatogram, chromatography resin, column equilibration. That's what happens with binding buffer. So one has to equilibrate the column first before loading the sample. There's column packing as well. Basically, the matrix can eventually be packed in glass columns also. Okay. One talks frequently about column volume, which is the complete uh, volume occupied by the in the chromatography bed. When we talk counter ions, etc., we will look eventually in the ion exchange chromatography or counter ligand, different types of chromatography. When we are talking about different types of chromatography, we will look at these terms in detail. There's something called dead volume. So, when we are talking constantly about chromatographic resin or chromatography matrix, those are basically different kinds of substances or compounds cross-linked with each other or packed together, but still the packing capacity or packing efficiency is not 100% definitely. So there will be gaps in between different kinds of when two compounds are cross-linked to each other, two compounds are present, still there will be gaps in between. Okay, how we were looking at agarose gels, polyacrylamide gels, right? So there were pores in between. So uh, basically, dead volume is the volume outside the packed chromatographic resin or chromatography bed. Okay, so it can be column dead volume or column chromatography system dead volume, and the dead volume contributes to band broadening. Another term is degassing, where one has to remove the dissolved air from buffers or solutions, because for gel filtration or SEC, we will eventually look at it in great detail in later slides as well as in the next tutorial as well, where this is very necessary so that it doesn't interfere with the uh, column packing and column chromatographic column resolution. Okay. Desorption is basically opposite of adsorption, where the bound solutes are eventually eluted off. It detaches from the matrix, interactions are weakened, and it elutes out. And then there's effluent, eluent, elution buffer, elution volume, elution time. There's flow rate as well, flow through, which is coming out eventually. Okay. Then there's hydrophobic interaction chromatography. These are different kinds of uh, terms you can come across, and each of these we will be taking care of in the later slides. Void volume we've talked about dead volume, void volume, the elution volume of solutes that do not enter the pores or interact with the resin or chromatographic matrix in question. So it passes between the particles in the packed bed. Okay. Then there's always a wash or wash buffer. Usually, suppose you want to purify a protein. You have first equilibrated the column, giving it the same conditions that the protein requires or the protein needs, or the pH conditions that the protein needs to maintain a specific charge, overall charge on it. But then the protein is bound to the matrix. But along with that, some other contaminants might also be bound to the matrix. But you have to remove, you need a pure sample, pure protein. So you have to remove the contaminants from the protein bound onto the matrix. Okay. So what one does is, always there are either more than one or two, three washing steps in between, where you are removing the non-specifically bound contaminants uh, s- and like leading to much pure or more pure sample. Okay. So there can be different applications of chromatographic techniques. Now, the different properties that are actually used to separate out uh, for different kinds of chromatographic techniques, charge, overall charge on the molecule of interest or on the analyte can be used in ion exchange chromatography or chromatofocusing. Chromatofocusing actually separates molecules based on the isoelectric point of the or rather the net, like how far or where exactly it moves in an electric field, where exactly the molecule of interest or macromolecular machinery moves or stays uh, in an electric field, but there's no movement at its isoelectric point. Also, there's the separation of samples based on the size or hydrodynamic radius, basically known as gel filtration or size exclusion. And there's the property of hydrophobicity can also be exploited, and the hydrophobic interaction chromatography or reverse phase chromatography can be used. And then there's ligand specificity, or based on the specificity of the ligands, different affinity chromatographies can also be used. Okay. Now, first let's talk about ion exchange chromatography. It actually takes advantage of the relationship between the net surface charge that the molecule or analyte has and the pH for a specific protein. Okay. Again, the interactions have to be reversible because once the specific protein of interest with the specific charge, net charge on it, is bound to the matrix, and the other uh, at, at the other point, due to change in the buffer conditions, the protein's interaction with the matrix weakens and the protein elutes out. This is the protein that we are interested in. This is our protein of interest. Okay. So it is actually the interactions are reversible between the charged molecules and the oppositely charged ion exchange media. These are actually controlled, mainly the pH is controlled to favor the binding or elution of the molecules and achieve separation. So, a protein having a no net charge at a pH equivalent to its isoelectric point will not interact with a charged medium. But at a pH above its pi, a protein will bind to a positively charged medium or an ion exchanger. So, we were looking at in the earlier tutorial classes as well, where if the pH is higher, that means the H plus content is less, right? OH minus concentration is higher. Now what happens is, if the H plus concentration is less, then eventually the NH, the N terminal or NH2 group, which exists as NH3 plus after donating its lone pair, will, due to lesser concentration in the lesser concentration of H plus in the surrounding medium, the NH3 plus gives off its H plus ion, thereby forming NH2. Okay, but COOH group already has donated H plus ion and it is existing as a carboxylate ion, right? CO minus. So the net charge on it is -1. Okay, net charge on it is a negative charge, right? But eventually, if the pH is lesser, H plus ions, concentration of H plus ions is more. So eventually COH will exist, CO minus will exist as COH, and NH2 actually now the lone pair on the nitrogen can eventually be given off to hydrogen, so that the N terminal portion exists as NH3 plus. Okay. Now, if at a pH above its pi, a protein will be negatively charged. Hence, the protein will bind to a positively charged medium, or a positively charged medium or positively charged matrix is also known as an anion exchanger because the protein is in its anionic form, right? And at a pH below its pi, a protein will bind to a negative, it will be bind, sorry, not behind. It will bind to a negatively charged medium or a cation exchanger because at a pH below the isoelectric point of a protein, the protein is positively charged, right? So eventually the cation unique form of the protein is getting exchanged out of the sample mixture. Okay. Various other types of binding can also occur, but these effects are really small and mainly due to van der Waals forces and non-polar interactions. Okay. So when we look at the theoretical titration curves of the protein, we were looking at the glycine titration curve, right, with two pi values. Hai na? So here, similarly, when like this particular thing shows how the net surface charge varies with the pH. Okay. So if this is the matrix, and the matrix actually has positively charged ionic groups attached to it. Now, a low ionic strength buffer is passed through it, and negatively charged molecules from the buffer uh, can interact with the positively charged groups on the matrix. Okay, this is what happens when we are equilibrating the matrix or equilibrating the column. Now, when you are actually applying your sample, or sample of interest, now these positively charged residues are bound to the matrix. So overall, the matrix is positively charged. Now, either your positively charged sample will be, your negatively charged sample will be binding to the positively charged groups on the matrix, or there might be some weak interactions between neutral molecules as well, or neutral entities as well, that can interact with the positively charged matrix here. Okay. So now what happens here is, during equilibration, this is what is happening. Okay. Now, when you have loaded your sample, so the oppositely charged proteins are binding to the ionic groups of the ion exchange medium, and uncharged proteins, or those with the same charge, definitely the ones which are positively charged, will be repelled and eluted out with the solvent, okay, or the buffer that is being passed through, or the mobile phase in question. So uncharged proteins, or those with the same charge as the ionic groups, will elute during or just after the sample application. So this is what is happening here. Now you have loaded the sample. Now this is your protein of interest, right? Now what happens is, now you actually have to take this protein of interest out of the matrix now. It is adsorbed onto the matrix. It has interacted with the matrix and bound to the matrix via electrostatic interactions, like charged interactions. Now what happens is, next you increase the ionic strength of the buffer in question. Basically, it should have a large number of negatively charged ions, okay, which can actually interact more with the positively charged matrix. Okay, eventually displacing your negatively charged protein molecules, or your negatively charged molecule of interest, and your molecule, this green one here, will eventually elute out of the chromatographic column, and if you will have your molecule of interest. Okay. So increasing the ionic strength displaces the bound proteins, as ions in the buffer compete for binding sites. These blue ones are competing with the protein for binding sites onto the positively charged matrix. Further increases in the ionic strength actually displaces the proteins that are more highly charged or are tightly bound. Okay. So, but again, before elution, as I was talking about, after equilibration, you have loaded the sample, then you have to wash the samples, so that these neutral charged ones, which you do not really need, uh, basically the contaminants, you have to remove those contaminants, or non-specifically bound contaminants which are bound to your matrix non-specifically. Those have to be removed, so that you only have the protein of interest, which is this green colored complex in question now, like in the picture that has been shown. Okay, so that is washed out, and eventually, even after your elution also, one needs to wash the column properly. Okay. Now this is the same thing. Resolution of the matrix, or resolution of the column, that we were talking about, is the distance between the peak maxima, right, compared divided by the average base width of the two peaks. Okay. Now when we talk about elution volumes and peak widths, basically are dimensionless, and resolution gives a measure of the relative separation between the two peaks, and this overlapping ones means resolution is lower, in a very ideal case scenario, you actually have two well separated distinct peaks for a like in a chromatograph. But again, one thing that we should be very careful of is a single well resolved peak does not necessarily represent a pure substance. Okay, it can also have a series of components or a mixture of components which could not be separated under the given elution conditions. So just by looking at the peaks, you can have an idea, but then the samples corresponding to that particular peak, suppose peak A or peak B, has to be analyzed further via electrophoresis or via some assays or via activity gases, etc., to check the to check whether that is actually the protein of your interest or not, or actually the complex of your interest or not. Okay. Now, another thing that has to be taken care of is the particle size. It is actually a very significant factor in the resolution of a column, chromatographic column. And in general, the smallest particles will produce the narrowest peaks, okay? Like this, like really sharp and narrow peaks under if the elution conditions are really optimum and the column is well packed, okay? Now, there can be gradient elution or stepwise elution, okay? So in gradient elution, what happens is when start when you are actually starting the purification of a completely unknown sample, suppose a protein in question. So many components are there in the sample mixture, right? And many components will be bound to the column as well and will be eluted differently, okay, based on their interactions with the matrix as well as interaction with the buffer or the mobile phase, okay. So for that, a gradient is given where we don't really know at what kind of, suppose we looked at ion exchange chromatography, right? So we don't really know what kind of ionic strength will eventually elute out our protein of interest or our molecule of interest. So for that, the range of basically from 0% to 100% um, ionic strength of the buffer, elution buffer is given so that it your like our protein of interest, our molecule of interest can elute out at 50% of the ionic strength, okay, 50% of the highest ionic strength, right? Or it can even elute out at 80% of the highest ionic strength. So that has to be optimized for, and it is different for each type of protein of interest or your molecule of interest, okay. But what actually happens here is during gradient elution, this is the initial steps where you are optimizing the purification of your sample of interest. Okay, when we are talking about protein, so eventually have to you end up making a lot of buffers for gradient elution. A larger, comparatively a larger volume of buffer is required. But when you have optimized, suppose you know that your protein elutes at 50% of the highest or 50% of um, 1 molar salt, okay, or at like it elutes at 500 millimolar of salt. Now you already have optimized this condition, so you needn't the in the next rounds of purification of your protein, what you can do is you can directly put in 500 millimolar KCl salt and elute out your protein of interest, okay. So when an ion exchange separation has been optimized using gradient elution, changing to a step elution actually definitely it is speeding up the separation times, and also it is reducing the buffer consumption while retaining the required purity level, right? And step elution can also be actually used for group separation in order to concentrate the proteins of interest and quickly remove them, quickly remove the proteins of interest or multiple proteins of interest from unwanted substances. Okay, so what actually happens eventually is the target protein or the multiple proteins are eluted in an enriched and more concentrated, more pure form. Also, step elution can be used to remove the contaminants, like basically the ones, the molecules which are not of your interest, but still in the sample, but still it's there in the sample mixture. Step elution is used to remove contaminants by choosing conditions that maximize the binding of the contaminants and allow the target proteins to pass through the column. So care must be taken to ensure that the binding capacity of the column is sufficient to bind all the contaminants. So we looked at ion exchange chromatography. Now let's look at hydrophobic exchange chromatography. This actually exploits the ability of a strong interaction between a hydrophobic group attached to the column or column matrix or column resin and the hydrophobic patches present on an analyte such as a protein. Okay. So hydrophobic patches are usually folded, like when we are talking about a protein. Hydrophobic residues, if it's a polar environment, hydrophobic residues are actually well folded and well protected from the polar environment and usually present on the inside. But when the polarity is actually reversed or it changes, what is happening here is eventually the hydrophobic patches are exposed outside and the structure reverses or denatures. Okay. Now, hydrophobic interaction chromatography also actually mainly uses the salting in and salting out principle. What happens is addition of low amount of salt to the protein solution results in the displacement of bonded water molecule with an increase in protein solubility. This is called salting in effect. Okay. Now, when there's more amount of salt present, then the water molecules which actually shield the protein side chains are displaced, are removed completely, and the hydrophobic patches are actually exposed out on the protein surface, which actually leads to protein precipitation because the interaction of well-folded structure is eventually getting denatured. Okay, decrease in protein, and this results in decrease in protein solubility. So, more amount of salt is leading to salting out effect. Okay. Now, the phenomenon of salting out is can be optimized or regulated so that addition of salt induces exposure of hydrophobic patches on the protein, but it does not cause precipitation or aggregation. Exposure of hydrophobic patches on the proteins actually can facilitate the binding of protein to some non-polar ligand which is attached to the matrix. This is actually what results in hydrophobic interaction chromatography. Now, if the concentration of salt is decreased, the exposed hydrophobic patches on the protein reduces the affinity towards the matrix and your protein of interest eventually elutes out because the interaction with the matrix is decreased, has decreased. Okay. So this is what is happening here at high salt concentration. The protein is bound to the matrix because the hydrophobic patches are eventually getting exposed. But eventually when the salt is reduced, salt concentration is reduced, the hydrophobic patches are less exposed and hence the interaction with the matrix reduces and the protein elutes out. Okay. Now let us look at some of the questions, sample question. So which of the following is a physical method of cell disruption? So the answer is option C, because this is a chemical, this is permeabilizing, this is alkali treatment. So we are talking about a physical method of disrupting cells, which is sonication. We looked at osmotic shock method, which can be employed in cases where the cells actually lack a cell wall. Okay, so the option will be option C. Now this is a question regarding ultrasonication, where what is the frequency uh, required to disrupt the cells using sonication? The answer is option B, 20 kilohertz. Okay. Now again, this next question deals with sonication of cells. When we are sonicating cells suspended in a liquid medium, so we usually do it on ice to prevent thermal denaturation. Actually, due to the cavitation and the bubble formation and the explosion of the bubbles, what happens is there's a lot of heat is generated. That's why uh, always sonication is done while keeping the samples on ice. Okay. So that because the molecule of interest or the analyte that you eventually wish to purify or isolate can be susceptible to higher temperatures. Okay, so we have to keep the sample on ice, and then we looked at the titanium probe that is eventually kept inside the cell suspension to to disrupt the cells. So we have to keep it on ice so that we prevent the thermal denaturation of the intracellular macromolecules. Okay. Lipid saponification is a result of treatment with, so we looked at lipid saponification. So treatment with alkali leads to lipid saponification. So in general, what uh, saponification is basically soap formation. So when a lipid, triacyl glycerol is an example of a lipid, it is getting hydrolyzed with a strong base or alkali. Sorry, the process is called saponification. And what happens, what eventually you get in saponification are glycerol and fatty acid salts. Okay. So what is happening here is NaOH, usually in saponification for alkali, like for disruption of the cells, sodium hydroxide is used with saturated fats to produce hard soaps. Uh, KOH is used to usually interact with unsaturated fats to produce softer, more liquid soaps. Okay. Basically, saponification is soap formation. Okay. Now, when we talk about enzymatic digestion of yeast cells would result in the formation of what kind of entity? This is option B, spheroblast. So we very recently looked at hydrophobic interaction chromatography. So what happens when we add a low amount of salt, which facilitates the solubility of a protein, basically detachment of the protein from the matrix. So this is also known as what kind of phenomenon? Which is option B, salting in. Basically, you are adding more amount of like more amount of salt is exposing the hydrophobic patches, and the protein is interacting with the matrix. But when you are increasing, like decreasing the amount of salt, eventually the hydrophobic patches are less exposed, and the interaction with the matrix decreases, and the protein eludes out. Now the pi of a protein is 6.5. So which of the following buffers can be used so that the protein of interest can bind to a cation exchange column for purification? Cation exchange column for purification means if the pi is 6.5, cation exchange column for purification means the protein should be positively charged, right? So it should have a pH less than 6.5, less than the pi of the protein. Then only the complete net charge on the protein will be positive. Okay. So a phosphate buffer with pH 5.5, which is less than the isoelectric point of the protein, should be used for cation exchange chromatography. So now this is an example of an ion exchanger. An ion exchanger means the matrix is positively charged and negatively charged molecules or molecule of our interest will bind to the matrix. Okay. So, like nucleic acids, etc. For that, nucleic acids are negatively charged, right, due to their phosphate groups. So, like for tRNA, tRNA purification, tRNA isolation, all these, diethyl aminoethyl DEAE matrix is used uh, to separate out nucleic acids. Okay, this is basically positively charged so that negatively charged substances can bind to the matrix. Now, a mixture of four proteins with their charges in a phosphate buffer of pH 7.4 uh, has um, these charges. Okay, and the buffer has a pH of 7.4. Now, which protein will bind strongly to the matrix of an anion exchange column? So the matrix of an anion exchange column is what? Positively charged, right? So out of all these charged residues or charged entities, the one with the highest negative charge will actually bind to an ion exchange column, which is positively charged. The matrix is positively charged, and the molecule that will bind to it or interact with it will be negatively charged. Right? So the correct option will be option D. Okay. Next is which of the following property of a protein is exploited in gel filtration chromatography? This is the hydrodynamic radius or the size. So out of all this, the answer is option C, surface area. Now, when we talk about the distribution or the theoretical plates in a chromatographic column or chromatographic matrix, so the question is a chromatographic column has a length of 100 centimeter with a 1 centimeter height of the distribution plane. So how many distribution planes are there in that particular column. Okay, if the complete column length is 100 centimeter and each distribution plane has a height of 1 centimeter, then it will eventually be complete length divided by the height. Okay, so the answer will be option C. Basically, 100 centimeter is the complete length, and eventually each plate on the matrix is of 1 centimeter height. Okay, so how many planes are there, or how many plates are there on the column? That will eventually be divided by the height, I mean the length, full length of the column divided by the height of each plate. So the number of distribution plane in a column is actually controlling two parameters. So if the number of distribution plate will go up, then it will allow the analyte to travel for longer period of time, and it will increase the distance between two peaks. Basically, it is uh, increasing the resolution. And as the number of distribution plane will go up, it will also reduce the width, like make the peaks narrower and sharper, and the width of the base of the peak will reduce. As a result, the peaks will be sharper. Okay. So as the number here, this is a representative example of a chromatogram. So as we see that as the number of plates are increasing, the peaks are narrower and sharper. But for n is equal to 10, for number of plates being 10 in a column, in a matrix, uh, the you see a huge peak broadening or band broadening here. This actually leads to extremely bad resolution and extremely bad separation of the uh, sample or solutes in a sample mixture. Okay. Now, the distribution coefficients of x, y, and z are 1, 5, and 10 respectively. And this mixture of x, y, z are actually injected into a column. So what is the order of their elution? So the higher the distribution coefficient or KD value of a particular solute, the higher will be its interaction. Okay. So the lower the value of KD, the lower will be its interaction with the matrix, and hence it will come out of the matrix much faster and much earlier than the other solutes in the mixture, which are interacting much more strongly with the matrix. Okay. So 1, 5, 10. In the same order, the one with the lowest KD will elute out first. Okay. So the order of the elution will be 1, 5, and 10 respectively, right? So it will be option C, X, Y, Z. Now, when we are talking about cell disruption using bead milling method, okay, and release of the products, so what kind of kinetics is followed? Usually, what happens is the cell disruption in a bead milling and release of the product follows first order kinetics, okay, where Cmax is the concentration of the product that is actually released from a, or rather the intracellular content that is released from a given amount of cell suspension. And C here is the concentration of the content released at a particular time t. And this constant here, K, um, actually depends on the type of uh, apparatus that is being used, what is the size of the beads that is being used for disruption, and how, like, basically how much amount of the bead has been loaded in the cell suspension. Okay, and how much speed, like with how much speed you are agitating the suspension, as well as the temperature as well. Okay, so all these different conditions actually affect how fast or how efficiently a cell is disrupted using bead milling method. Okay. So basically the question asked what kind of kinetics it follows. So it is first order kinetics. Okay, option A. Now, which of the following equation describes the dynamic pressure experienced by cells in a high pressure homogenized? So it's half rho v square. Rho is the density. Okay, and v is the jet velocity, and overall dynamic pressure is Ps represented here. And so the option will be option A. Okay. So that's it for today's tutorial. We'll meet again the next tutorial. Okay. Next week, as in. Thank you for joining. And next week we will have the usual session on Saturdays. Thank you for joining. Thank you.